Welcome back to the Cell and Molecular Biology course. I'm your course instructor Iniyatindra. In the previous video, we saw a brief history of microscope, followed by how a light microscope works, and then how to view a slide under a microscope, followed by the types of light microscope. In this video, we will see the key differences between light microscopy and electron microscopy. Microscopy has revolutionized the way we study cells and cellular structures. Two key types are light microscopy and electron microscopy. Most of us have used a light microscope in school to observe cells or tissues. It's easy to use and it will allow us to see basic structures using visible light. However, when it comes to much smaller details, like the fine structure of a cell or cellular organelle, we need something more powerful, the electron microscope. Unlike the light microscope, it uses electron beams and requires specialized labs to operate. In this presentation, we will compare these two important tools and their unique capabilities. By the end of this video, you will be able to understand the fundamental principles of light microscopy and electron microscopy. And then you can compare the magnification, resolution, and sample preparation requirements of both techniques. And then you will be able to identify the key applications and limitations of each type of microscope. When studying a cell, which would you prefer to use? A light microscope or an electron microscope? What factors would influence your choice? By the end of this video, you will get to know when to use light microscopy technique and when to use electron microscopy technique while studying a cell. Let's begin with the difference between light microscopy and electron microscopy. To begin with the basic difference, let's see the illumination source. As the name says, the light microscopy uses visible light to observe the samples, while the electron microscopy uses a beam of electrons. Let's see the basic working principle. Light microscopy and electron microscopy are two fundamental tools in biology, each based on different principles. Light microscopy uses visible light to illuminate a specimen. The light passes through or reflects off the sample, and lenses magnify the image. This will allow us to see structures in living cells and tissues. It's widely used in labs for basic observation. Electron microscopy, in contrast, uses a beam of electrons instead of light. Since electrons interact differently with materials than light, this method allows us to see much smaller structures. The specimen must be in a vacuum, and the electrons either pass through or scan its surface, creating detailed images of cellular structures. While it offers a greater detail, electron microscopy requires a more complex preparation of samples, and we will see the steps involved in detail further in this video. Light microscopy and electron microscopy differ fundamentally in the type of lenses they use. In light microscopy, glass lenses are employed to bend and focus light, allowing us to magnify the image of a specimen. These lenses work by refracting light as it passes through them. This will enable us to see the objects that are otherwise too small for the naked eye. In electron microscopy, however, electromagnetic lenses are used instead of glass. These lenses control and focus a beam of electrons. Since electrons are charged particles, electromagnetic fields can guide them in much the same way that the glass lenses bend light. This allows electron microscope to produce highly detailed images of much smaller structures than light microscope, although they require more complex technology to operate. Let's see the magnification power of both light microscopy and electron microscopy. Light microscopy and electron microscopy differ significantly in their magnification. In light microscopy, magnification is achieved through the use of glass lenses that bend and focus light rays, making the image of a specimen appear larger. The total magnification is the product of the objective and eyepiece lenses, typically allowing us to see structures that are up to 1,000 times larger than their actual size. Electron microscopy, on the other hand, uses a beam of electrons and electromagnetic lenses to magnify the image. Since electrons have much shorter wavelength than visible light, electron microscopes can magnify objects up to millions of times their original size. This allows us to observe much finer details of a specimen, such as the internal structure of cells or even individual molecules, which light microscope cannot resolve. Resolution. Resolution is the ability of a microscope to distinguish between two closely spaced points. And this is where light microscopy and electron microscopy significantly differ. In light microscopy, the resolution is limited by the wavelength of the visible light. This means that while you can see small structures within cells, there is a limit to the detail that can be observed, usually around 200 nanometers. Electron microscopy, however, offers much higher resolution. Instead of light, it uses electrons, which have a much shorter wavelength. This allows electron microscopes to distinguish between much smaller structures, revealing details down to the nanometer level. Diffraction. Diffraction plays a key role in the functioning of both light and electron microscopy. In light microscopy, diffraction occurs when light waves pass through or around small objects like cells. This limits the microscope's ability to resolve fine details as light waves spread and overlap when they encounter very small structures. This diffraction limit restricts the resolution of light microscopes as light waves spread and overlap when they encounter very small structures. In electron microscopy, electrons, like light, also behave as waves. However, because the wavelength of electrons is much shorter than that of visible light, diffraction is less of an issue. This allows electron microscopes to overcome the diffraction limit faced by light microscope, enabling them to resolve much smaller structures and provide more detailed images. Depth of field. Generally, light microscopy has a greater depth of field meaning a larger portion of specimen in focus at the same time. While electron microscopy, on the other hand, has a shallower depth of field, especially transmission electron microscope has a shallower depth of field, making it more difficult to focus on all parts of a thick specimen simultaneously. Let's see the major differences in terms of sample preparation between the light microscopy and electron microscopy. The complexity is relatively simple in light microscopy, while in electron microscopy, the sample preparation is complex and also time consuming. The techniques involved in light microscopy sample preparation includes staining, such as gram stain and Gemsa stain, and also simple mounting, like mounting the sample on a slide. And then it may also require fixation, such as heat fixation. Based on the requirement, these techniques can be followed. But electron microscopy, on the other hand, has complex procedures, including fixation, dehydration, embedding, sectioning, and staining. Let's see the steps in detail. Fixation. Fixation involves preserving the sample structures. Example using chemicals like glutaraldehyde. The next step, dehydration, involves removing the water from the sample. Embedding. Embedding involves infiltrating the sample with a resin for support. Then comes the sectioning, cutting the sample into very thin sections, ultramicrotomy. Then later comes the staining, usually heavy metals, example osmium tetroxide, uranyl acetate, lead citrate. All these are used to increase contrast. The samples used for considerations. That is, the samples used to observe under light microscopy can be a living specimen or a non-living specimen, while samples used to observe under electron microscopy cannot be a living specimen. Light microscopy typically produces 2D images as the thin slices of specimens or live cells are observed from a top-down view. However, with advanced techniques like confocal microscopy, 3D reconstructions of cells and tissues can be created by combining multiple 2D slices. In case of electron microscopy, transmission electron microscopy produces highly detailed 2D images by passing electrons through a very thin specimen, while scanning electron microscopy produces 3D images by scanning the surface of the specimen with electrons, showing detailed surface structures in a three-dimensional view. In case of color, light microscopy can show images in natural color, especially with stains and dyes that highlight specific structures in the cells. Fluorescent microscopy allows even more vibrant and specific color differentiation by labeling parts of the cell with fluorescent markers. In case of electron microscopy, it produces grayscale black and white images as it captures interaction between electrons and the sample rather than visible light. However, false color can be added digitally for interpretative purposes in scientific presentation. The type of image obtained. In light microscopy, the images are usually less detailed but can capture living cells and their dynamic processes, showing basic structures like the nucleus and organelles. Light microscopes are especially useful for observing the overall organization of cells or tissues, while electron microscopy produces highly detailed, high resolution images showing fine structures like organelles, membranes, or even viruses. Transmission electron microscopic images provide internal structure details, while scanning electron microscopic images show surface textures and morphology with a 3D effect. Now let's see the different types of light and electron microscopy. The types of light microscopy includes bright field microscopy, which is the most basic form of light microscopy, and phase contrast microscopy, fluorescent microscopy, and confocal microscopy. The all these types are discussed in detail in the earlier video, and the types of electron microscopy includes transmission electron microscopy, scanning electron microscopy, scanning transmission electron microscopy, and cryo-electron microscopy. Each type of microscopy is tailored for specific purposes, with light microscopy excelling in live cell imaging and electron microscopy providing unmatched detail for studying fine structures at molecular level. Now let's see the cost and availability. Light microscopy is affordable and is widely available, while electron microscopy is expensive and it requires specialized labs. Building on the differences we have just covered, let's now explore how both light and electron microscopy are applied in various fields. Light microscopy, with its ability to magnify up to around 1000x, is essential for observing live specimens. It's widely used in biological research and healthcare, allowing scientists and medical professionals to study living cells, tissues, and microorganisms. It's particularly helpful in diagnosing diseases such as infection, cancer, and inflammatory conditions by enabling the examination of cell morphology and tissue structure. In education, light microscopy serves as a fundamental tool for students to explore the world of biology and understand cellular structures in a hands-on way. Electron microscopy, in contrast, offers far greater magnification, reaching up to millions of times, making it indispensable for observing structures at the molecular and atomic levels. This allows for the detailed examination of viruses, organelles like mitochondria, and even individual atoms. In fields like material science, electron microscopy enables the analysis of the composition and defects of metals and polymers, which is crucial for developing new materials. In medicine, it aids in understanding the intricate details of cellular damage, helping researchers to investigate diseases at a molecular level. Additionally, electron microscopy plays a vital role in virology, providing insights into the structures of viruses and the interaction with host cells. Both light and electron microscopy provides unique insights, making them essential tools for advancing research in biology, medicine, and material science. Now let's look at the advantages and limitations of light and electron microscopy. Light microscopy is useful because it allows us to observe living samples like cells or small organisms in real time. It's easy to use and affordable, and the sample also doesn't need much preparation. However, it has its limitations. It can't zoom in much, so we can't see tiny details like viruses or the structure of small cellular organelles. Electron microscopy, on the other hand, gives us much higher magnification and resolution. It provides detailed views of subcellular structures. This makes it perfect for studying viruses or cell parts that are too small for light microscopes, but it comes with some limitations. It's expensive and it requires special training to use, and the samples need to undergo a lot of preparation. Also, electron microscopes can't be used for living samples because the process can damage them. So, light microscopes are useful for looking at living things, while electron microscope gives us incredible detail but can't look at a live sample. Let's summarize the points we learnt in this video. Light microscopes use visible light and lenses to magnify objects, while electron microscopes use beam of electrons. Electron microscopes offer significantly higher magnification and resolution than light microscopes. Light microscopes are generally simpler, less expensive, and can be used to observe living specimens, while electron microscopes require complex sample preparation and cannot be used to observe living specimens. However, the choice of microscope depends on the specific research question and the nature of the sample. Microscopy, a powerful tool. Microscopy plays a crucial role in various scientific fields, from biology and medicine to material science and nanotechnology. In this video, we learned light microscopy and electron microscopy in detail. With that, we could conclude that both light and electron microscopes offer unique advantages and are essential tools for scientific research and discovery. The advancements in microscopy technology continue to push the boundary of our understanding of the micro and nanoscopic world. Thank you for watching. Hope this video was useful. See you in the next video.